Hello, and welcome to today's lecture, featuring introductory information on various neuroimaging methods. Cognitive neuroscience seeks to measure the brain activity elicited by different behaviors and cognitive processes. On the left, you see a stimulus, and on the right, you see three different formats to present and quantify brain activity associated with the stimulus. This is the goal of neuroimaging. Broadly, we know we're measuring activity in the brain, but each imaging method records a different type of signal, and each method has its pros and cons. There are many different flavors of neuroimaging. Generally, they rely on hemodynamic or electrical activity. Positron emission tomography uses a radioactive tracer which emits recordable gamma rays corresponding to recruitment of blood to different brain regions. This takes place over the course of 1 to 40 minutes. This method is good for locating receptor sites in the brain. Functional magnetic resonance imaging measures functional activity in the brain, rather than structural, which an MRI measures. Inference is made regarding the oxygenated blood being recruited to certain areas of the brain during some events and not others, with the assumption that increased blood flow represents increased activity. Functional near-infrared spectroscopy detects function only. This detects changes in concentrations of oxygenated and deoxygenated blood via the absorbance of near-infrared light. This is most useful for cortical activity, not subcortical, since the device relies on light, and this light can only penetrate the scalp so deeply. Magnetoencephalography uses magnetic fields given from electrical current in the brain, which can be measured much like electrical activity in EEG. However, these are less obstructed by the scalp and skull. Electroencephalography measures electrical activity emitted from postsynaptic activity. This is observed as it combines together on the surface of the scalp. This is just a broad overview of these different methods. Today, we're going to function on MRI, functional MRI, and EEG. Now, these methods all measure changes in blood flow and electrical activity, with each giving a different picture of what is happening in the brain from a different perspective. Because of the nature of how each method works, they each have separate pros and cons we'll discuss as we cover each method specifically. But on a simpler method, what are we actually observing? At the most simplistic level, brain activity refers to firing neurons, where the electrical signals produced at a neuron which has reached its action potential are sent to every neuron that cell synapses onto. This creates a constellation of electrical activity that is in constant flux. But most human neuroimaging methods do not look at the activity of single neurons. Rather, brain activity is measured through biophysical indices that are coupled with this firing. Let's talk first about how EEG does this. EEG measures chains of neurons firing together. Specifically, in the cortex, apical pyramidal neurons are arranged in parallel. When a neuron fires, the concentration of intracellular and extracellular ions change, creating an equivalent current dipole across the firing chain with a measurable electric field. This dipole sends a summed signal towards the skull, which then combines with other equivalent current dipoles to create the signal we observe with EEG. In other words, neuronal activity produces small local currents due to the influx and release of charged ions, with excitation from presynaptic neurons creating an influx of positively charged ions into the cell, whereas the extracellular space becomes negatively charged. The action potential travels down the neuron where positive ions leave the cell, making the extracellular space positively charged. In other words, these chains of neurons form a long signal generator, with one end being negatively charged and the other being positively charged, due to the cellular mechanisms involved in action potential generation. Now, magnetic resonance imaging can image both structural and functional aspects of the brain, using similar principles for different molecules in the body which exhibit a magnetic field. MRI focuses on hydrogen atoms for structural purposes, while functional MRI focuses on hemoglobin. Let's start with MRI. When a molecule has an unpaired proton, it creates a magnetic field, similar to those in a bar magnet or a compass, with north and south being the opposite poles. Normally, protons are randomly spinning. but in the homogeneous magnetic field of a scanner, protons align. Because the proton has a magnetic field, it can be influenced by external magnetic fields. In the magnet, once the protons are aligned, they are knocked over by another magnetic field. Then, this additional magnetic field is removed, and the atoms return to their homogeneous orientation originally created by the field of the magnet. As the protons dephase, or return to normal, they release energy in this process, and this ends up creating an intensity value affecting the MR signal. How does this work in our body? Different types of tissue have different concentrations of water molecules, and these molecules have their own magnetic spin. MRI uses the hydrogen in the water molecules to create its images. So remember, these molecules are magnetically oriented in identical fashion and bumped by an additional magnetic field. When this field is turned off, the hydrogen protons release this energy at a radio frequency which can be detected by the MR scanner. The differing amounts of energy released vary between tissue types, causing the multicolored contrasts we see in this image here. Now, functional MRI still uses magnetism and even the same scanner. But now we're looking at the blood. The fMRI signal is dependent on regional changes of blood flow brought to the brain through its neurovasculature. Now, blood contains two forms of hemoglobin, oxy and deoxyhemoglobin. Similar to water, the unpaired protons of the deoxyhemoglobin create a bar-like magnetic field that is attracted to nearby magnetic fields. As blood is brought to different portions of the brain, this change in overall magnetism can be detected with the MR scanner, and it is this change in blood flow which is our proxy for brain activity. This is our signal of interest for fMRI. Following neural activity, two processes happen in parallel to increase the amount of oxygen in that area. Oxygen metabolism lowers available oxygenated blood and produces a temporary increase in deoxygenated blood as oxygen is used by cells. Local cerebral blood flow then causes a large influx of oxygenated blood and a slight decrease in deoxygenated blood. Together, these changes in hemoglobin due to oxygen metabolism and cerebral blood flow cause a net increase of oxygenated blood and a slight decrease in deoxygenated blood. Due to the different magnetic properties of oxy and deoxygenated blood, MR is able to use magnetic fields to figure out the ratio of deoxygenated to oxygenated blood. This ratio represents our signal of interest for fMRI, the bold signal, or the blood oxygen level dependent signal, which will be discussed in more detail later in this semester. To summarize, EEG measures electrical activity generated in the cortex. The source of this activity is current flow from pyramidal neurons, many of which have to be active in a parallel structured manner to create a current dipole with a measurable field. MRI examines the tissue concentration of water molecules. These molecules are put in a homogeneous magnetic field and aligned. Once aligned, they are hit with another type of energy. When this is turned off, energy is released and measured from the water molecules. As the concentration of these molecules changes across tissue, so does the signal they emit, producing the different contrasts that you can view in structural MR images. For functional MRI, blood flow is the important measure. As the amount of oxygenated and deoxygenated blood changes, so does the overall magnetic field. This signal is used as a proxy for brain activity, since it is assumed that this blood flow is associated with neurons requiring more energy from becoming more active. This is the end of this lecture. Don't forget any remaining module activities.